you guys here, both of you. And well, let's thank them and enjoy the last minutes with them.
Okay, so well, before we start, we'd like to thank you because for us this was an experiment. So we don't know if the experiment worked, but we learned a lot of things by doing it. That you were our exactly. I think we learned a lot of things doing this experience, and I think you are a unique group of people in terms of many factors. So it was really a pleasure for us to be with you doing this. No, really, thank you very much for this. But I ruined everything. And why there is this? Okay. And okay, so what we do today is to have the last discussion about the two papers that were in the folder. So the idea is to start as usual by doing a group discussion. So hopefully each of you have read these papers, or one of them. But I think it's good if you divide in the usual. I don't remember if there are 4 or 5 because there are more people here than usual. So I'm. Yeah, usually there are more people there, but you can divide in the usual or unusual 4 groups. And the idea of this is always to, since in this case these two papers are really about a controversy, so they really disagree. So try to make a case for each of them, discuss what do you believe between the two, why you think one is right or wrong. The limitation, yeah. Exactly, so the limitation of both the papers and also whether you think that the discussion that is in this paper is the discussion we could have, or we should talk about something else still related to ecology. And so, yeah, dividing groups, 20 minutes, okay? 5 more minutes? Because I think you are arriving to a great point now. No, I'm joking. 5 more minutes. I go to say. Okay. E si sta incazzando. Perché lei gli ha... Ok. Okay. I already have an idea of which group should start. I have an idea of which group should start. Do you want to say that? All right. So, who wants to start? Yes. Great.
Hi guys. We were discussing about mostly about the big paper, the 10 pages one, 11 pages one, and the way that they defined the interactions. Because when we talk about interactions, we talk about mostly the individuals that are interacting. And here they classify a positive interaction in a way that this interaction is enhancing the diversity of the community. And this is something that is, for me, is a big jump that you do in an interaction. Like they talk about a plus-minus interaction in a way that a plus-minus interaction is a positive interaction, because in a big scale you enhance the diversity of the community, and they do this in experiments that just do a pairwise experiment. So you just take two species and put them in a petri dish, and like the results of growth rates, you do extrapolation to say that this is going to enhance or not the diversity of the community, and you are not seeing the whole community, you are just seeing a pairwise. And you, for me, is too much to do this jump. And who wants to talk?
Yeah. It's because it's a complex system. You are in an ecological system, and you have information about pairwise interactions, and then you go to an ecosystem with a lot of individuals, and then you say, oh, this grows the diversity, but you just know about pairwise interactions. You don't know how these interactions will happen when you have like 100, 1,000 of interactions going on at the same time. So you cannot go just bottom up in the complex system this way.
For biologists, was very hard to see parasitism as a positive interaction. Just this. And
I mean, to kind of try to provide a counterpoint to this idea, I think that in some sense, I think the main claim that we would want to refute, if we want to say that these interactions are not sufficiently described pairwise, is that you need to show that interactions are kind of almost never local, because if the interactions are happening locally, I think it's somewhat reasonable assumption, right?
What do you mean locally? In space.
Locally in space.
So it's
because just in very simple situation, you take like a piece of volume and you see the kinds of what is the mechanism of interaction actually. I think that because what could be happening So let's imagine some hypothetical situation where all of these individuals are kind of excreting things into the environment, and then within the environment there can be chemical reactions that change the nature of these metabolites, and then this resulting thing can provide a different effect depending. Then I think you're looking at a very complex situation where you're looking at things that are irreducibly higher order interactions. But otherwise, in some sense, you would imagine that the community is going to assemble itself from the pairwise interactions. It's just some complex game that they're playing where you can have like this cycle of inhibitions and do some kind of global stabilizing effect, something like that. But I think it would be still well described by pairwise interactions. Sorry?
No, that's your completely well described by pairwise.
Yes, well described.
Other comments? Yeah. Sorry.
I didn't mean to be like, sorry. It's because I don't agree because in the paper they say they have like one paper showing that if you change the conditions and if you put more other species or increase the diversity, this relations change. So I think, for example, we can have like a positive interaction when we are both of us, but when we put more people, we start to compete. So I think it's a good starting point to look at the pairwise, but when you interact with more species, maybe it's not what you're gonna see, you know? That's why I think it's not like so well represented by the pairwise. And also the environment, not only the number of species, but also the other conditions in the environment can change the interactions.
Yes.
So I think it was clear to everyone that the one of the problems is the definition of what's a positive and a negative interaction. And I think that it's important to reflect about what we want to do with those definitions, because what's the meaning of that? If you just want to know that species A and species B together, both of them compete or predate, I don't know. But from especially the science article, I think there is this idea of trying to scale it up to things like the stability of the community, the diversity of the community, which I think is indeed what ecology tries to do with this interaction approach. Maybe it's different if you think about the idea of doing biochemical controlling of some organism. So maybe this, knowing the exact interaction, if it's robust or not to the changes in the environment, for that single thing, I think it might be important. But if you want to know how the community as a whole will behave, I think you have to take in consideration how those interactions change with the background, and this more complex thing that's not necessarily pairwise. Some interactions that are very pervasive and you are not catching. So I think there should be a discussion about what's the point with doing these classifications of interactions.
One consideration is that this debate whether doing yes. This idea of asking whether doing bottom-up research is meaningful, it's a big deal. So the idea was...
Bottom-up is exactly these things of...
Yeah, so there is... So, for example, so there are many groups, including the one that I am, in which that think have taken let's say, have tackled the challenge of trying to understand how the community diversity using this bottom-up approach, which means that you believe that pairwise interactions are the most important, and then you can use these, the outcome of pairwise interactions to infer the outcome of, for example, three-way interactions. And there is the assumptions that higher order interactions, so those that are mediated by other species, are not important, which is a huge assumption. And then there is this whole other field that it's more a top-down approach that you say, okay, and this responds to the necessity, I would say, you have to think about it as studying communities. It's really painful, and so let's see if this approach works. This is a way to tackle the complexity and the diversity. Then you can go to the other spectrum. You can consider a top-down approach. That is, for example, when you do microbiome research, you sample, you sequence everything, and then you're there saying, well, and now? So here, the main difference is that, so you can imagine a spectrum in which you have tractability on one side and complexity on the other. And here you can try to understand mechanisms. On the other side, you get only correlations. Here you lose complexity. On the other side, you retain complexity. Long story short, it's a balance, and you can think about looking at both and maybe come up with syntheses or ideas to jump from one side of a spectrum to the other.
Yes, but I agree, and I think that in some sense you can ask yourself, what is the minimal set of experiments that I'd like to do to predict the behavior of a large community, right? And of course you can say, well, I can study this in monoculture. Is it enough? Probably not. I can study this in pairwise. It's enough. I don't know, and so on and so forth. Right? So I think that, I mean, your question, it's really, I think, central. Why should we classify interaction? I think we should classify interaction if we believe that interactions are the important parameters, pairwise interactions, the important parameters in determining what happens at a larger scale.
About the idea that parasitism is a positive interaction, I agree that is a bit of a stretch. The only thing is that, I guess, as I told you before, the only thing is just thinking about the fact that someone is increasing the yield, so that's why there is a plus, and someone else is decreasing the yield. That's the, forget about the positive, I would say.
For me, the biggest problem is that they put this in the title. So when you read the title, you're like, oh my God, positive interactions are common, and you're like, oh my God, they're all mutualists or something. And when you read, you see that it's parasitism. You feel like, positive interactions, parasitism is like a clickbait.
Writing papers for science is mostly doing.
No, yes. So, no, I agree. I mean, there is one point that I think that one of the main problems there is using the word parasitisms. It's not really only the positive interaction. I mean, at the end you see, right? So because you see a plus minus, it's not really parasitism. It's not that one is parasiting the other, right? It's likely that I don't know, perhaps one possibility to have a plus minus is that in isolation the species grows first on glucose and then on acetate, but then in presence of the other only grows on glucose and the second grows on acetate, right? So what you see in that case is a plus minus, right? The first one doesn't grow on glucose. It grows in the presence of the others because there is overflow of acetate, and the first one grows less because it doesn't metabolize acetate because the other one is a bit. So it's a plus minus. It's parasitism. No. It's positive. Well, perhaps yes, in the sense that if you see from the perspective of the species that is between quotes parasitizing, it cannot grow on glucose, but in the presence of the other can grow on glucose. Right?
Yeah, so the idea, so this thing is that usually you define plus minus as parasitism. In this case, it doesn't mean what is usually meant. Yes. So that's the thing. So that's why if you think about it as both species survive, and one is actually doing better, that is why it's classified as a positive interaction. It's the definition that is off, probably. If we have in mind, I don't know, what's the name of the...
A parasite.
Yeah, like the typical parasite that we have in mind.
For me, it's like positive for who? Like,
Yeah, but
if one
No, I
is
agree.
minus, you are going to say that the interaction is positive, but positive for who, how, or...
yeah, but for instance, no, this is an excellent question. But for instance, you can ask, what is the total productivity of the community? So what is the total yield, right? And it could happen that the total yield is higher in the presence of a plus-minus interaction, right? So if you are thinking about the productivity of the community, the productivity has increased because of the presence of both, right? It's not what they address, it's a different angle.
No, but if you think about the fact, see, in the example that you were saying, if there is glucose, the species that grows only on acetate, let's say it can grow on acetate, in monoculture cannot grow.
Yes.
So it needs the other one to grow. So in this sense, this is how, okay, there is no judgment here. I'm trying to explain the reasoning behind how you're classifying these interactions. So because everything is related to monocultures, and if you can, and I think one of the points that one can take away from that paper is that many of the bugs that they addressed could not grow alone. So this is, for example, the starting point from saying, since I have so many bugs that cannot grow alone, and then when I put them in co-culture with someone else can grow, that's how you can think about these are all positive interactions. That's the type of reasoning.
Yep, something that we discussed was exactly this thing about the difference between microbial ecology and general ecology, because when we are talking about interactions in ecology, generally we are thinking about one-to-one relationship of an individual to an individual. And here, at the first time I read this paper, I was, oh, that's completely wrong, because parasitism is not a positive thing. But then thinking about the community as a whole, and generally when we are talking about microbial communities, we are talking about as a whole, not just punctual colonies. We are seeing that even with this interaction, the number of individuals that are increasing, and by somehow the number of different species are not decreasing. So in a sense, when you look, the big picture is a positive thing for the number of the community, and then we have the other interactions that maybe, you know, help even more. So
okay. I think that perhaps I'm still stuck in what Nicole said and about the definition, because it is another point in science where the investigators' view is in play, and we somehow transfer what is more epistemological to the things we are studying than are really trying to decide between two facts, because when you for example, in this case of how microbiological ecology, microbial ecology is done, you remove from the environment two things, and somehow you want to say something about that from which you removed.
But
so, and you do not really decide what you mean by positive, because you. I'll try and think again.
I don't understand if your point is against dichotomies, or it's a point against the possibility of investigating a complex reality by taking a piece and bringing it to the lab? Or it's something else?
I think it's I'll think again.
Okay.
I'll
Yes.
phrase.
Okay.
For me, one thing that was tricky about this article, this work, is like, yes, for me it makes sense. Like, if you look in the higher not dimension, but a higher scale, yes, this interaction could be positive in the community level, but they didn't measure this kind of thing. That's what bugs me. Like, they only measure the pairwise. So how can we have the discussion if they didn't even measure it? Like, for me, the conclusions they arrive, they are very strong for things they didn't measure. That's what annoys me.
Yeah, I think it's, it's again related to the bottom-up idea in some sense, or assumption, right? That you can extrapolate. Actually, it's interesting because both of the paper extrapolate, right? Because also the other, says actually mutualism cannot be present because we know that it's destabilizing and communities are stable, right? So I think it's interesting that both of them, even if they are reach different conclusions, they sort of are within this framework. I mean, the other point about, because this sort of emerged along the discussion about individuals and single cells. So you can do experiments where you look at the effect of other individuals on the growth rate of a single cell, okay? So basically you have these, are called microfluidic devices, which are basically allowed to put cells in a 2D layer. And you can have two different strains, which are labeled, so they have different colors. And you can follow the growth and division of, actually I have the, I don't know if, no. So you can follow the growth of these bacteria in time. I don't know if I find the right moment. Well, you can see a picture, not a, no, this is, anyway. So you see basically these bacteria growing and dividing, and so you can look at the growth rate of a single cell as a function of the cells that are around this cell, right? So that's something that can be done. So the fact that you measure these in communities, yeah, you see now that they're growing. Now they stopped. But you can really go down to the level of single cells. So it's not something that is just a collective properties, but doesn't have any ground into basic physiology. Yeah. Yeah.
A question for me is like systems biology, the way I see, the way I think that I understood, is something that studies the big picture, that this kind of network, these huge things. How is it really important to have these kind of very detailed studies for a single cell when you're considering a system? Like, can we actually? Is it reasonable to extrapolate these to the others? Can we jump scales? Does it make sense? I don't know. I was thinking. Like, I got it that it is a starting point. We must start somewhere. But it is really helpful.
I think no one believes that this is like the only starting point or the only approach. I think it's one approach, and if you think about it, it makes sense, right? So suppose that you want to, okay, let's forget for a second that all this interaction are environmentally mediated. You want to infer the network of interaction in a given community, right? How do you do this? Right? It's not obvious how to do it, right? And you can imagine to try to infer this network from complex data, which has its own limitation. Or you can say, well, I believe that somehow, I mean, I want to start what happens in small community first and then scale it up, right?
Yeah, the other thing that I can add is not just microbes. So we study plants like this. So I don't know, there is Jonathan Levine is competing plants. So, and it's the same type of approach. It's just that, again, it's a trade-off between complexity and tractability. And if you go towards complexity, you lose the ability to infer mechanisms because you're not manipulating it. So it derives from the fact that if you want to infer causal relationships, our way is to manipulate, right? You do an experiment, you manipulate, and then you can actually infer causal relationships. Especially for microbes, you can't really do it, or at least we're still trying. We still have to understand perfectly how to do it. So on the other side, you can say, okay, if I start with very small systems, where I can actually do manipulations and try to infer causal relationships, then I can ask myself, okay, how can I scale this up? And the scaling up is you know, one of these big problems and challenges. But yeah, so to me, it's always a trade-off between the things that you can say and the level at which you can say them. So either correlations or causation.
But to give another example, I mean, if you think what you do, like for instance, when you study pollinators, right? You collect data at the individual level, and then you extract, which is even a finer level than this, right? You collect data at the individual level, so you really see like which individual pollinator, what goes on an individual plant, and then from these networks of interactions between individual, you try to extrapolate things about the community, right? So I don't think it's something that is, as Martina said, is only for microbes. Yeah, I think it's in some sense, a standard approach, right? And if you think here, you have actually many more statistics, right? You can do many more pairs, many more conditions, etc. This doesn't mean that it's the right way to go. It just means that it's not completely hopeless.
About the separating something into bits, I am not opposed to that, but I am opposed to that when you try and do that and keep the same concepts you had, which are essentially global and macro concepts, and maintain them in a leaky and bad version in them. I think you should use new concepts and say, oh, I have this in this other scale, reducing the level of complexity. I see this, and I have a name for this. And then you forget about how you used to say, oh, lions and zebras have a positive negative interaction, and you simply face the problem that you are saying. It is a low order interaction, and I know that low order interactions are like this, and I don't try to say, oh, because what people want, as someone said, Ecology wants to do this push forward to try and globalize, but you should not in every title of every paper try and globalize results which are not global, because
Se anche fuori schermo puoi fare quello che vuoi.
Because in this case of I feel that in the paper that argues for the beneficial mutual relations being common, you mix a lot of characterizations of the community. You mix having a higher degree of general biomass or volume of the community, whatever is the metric you use, with the species level perspective, with the perspective of each cell with another cell. I think those things should be kept separate in order not to make those successive jumps which which are kind of faulty. I don't know. It bothered me a lot. The jump from what the paper shows to the title and the abstract. Sorry for the rain.
No, no. It's, no, yeah, I mean, I think like to sort of, I don't know, try to take the other, I mean, I think again, I don't think it's hopeless to try, I mean, it's how most of biology is done, I would say, that you have a very complex system, but then you try to isolate a piece, understand that piece, and then put it in context with the rest, right? And it's how most of biology works, and it has been successful in many cases, right? So,
Not just biology.
No, not just biology, but let's say, and it's true, like, yes, you can say I isolate two planets or a star and a planet in a solar system. That's, yes. No, but you don't, well, but that is observational, right? When you do experiments, when you manipulate, right? You manipulate a small part with the idea that it is an effect on something else which is fairly small and well-defined, and you can sort of forget about all the other parts, right? And then you can extrapolate, which I think it's a big assumption. I mean, sometimes it works, sometimes it doesn't work. But I don't think it's per se wrong, like trying to understand complex communities by studying simple communities, okay? I think that the connection, I agree with you that the connection is very far from being trivial, and it's really hard to scale this up. I mean, it's very hard to scale this up to 10 species, and it's impossible to scale it up to...
So the scale-up problem is also if you study a process at the small scale, I don't know, like variability in the distribution of species. And also if you, so scaling up is not just with communities, it's with spatial scale, with temporal scales. So in general, the scaling up problem is pervasive in science, and solutions are not clear. So it's just taking bits and bits, and then also science is kind of patchy in the sense that not everyone is doing everything, and so also connecting all the information is another big deal. But yeah, so to me these are problems that transcend microbial ecology. So these are problems that are pervasive in science. If you study something small, how much you can extrapolate to something bigger. There are concepts like, I don't know, scale invariance. If you find scale invariance, then you're happy because then you say, okay, if I study this at the scale, then I can really extrapolate. But how common is scale invariant? Yes, maybe in cauliflowers. And I don't know, other systems for sure. But so it's a huge problem for it is per se a problem. It's a pervasive issue.
So in some sense, I think that the question that it's important to ask is not whether it makes sense to do like experiments with such a small communities, but what kind of experiment should I do with these small communities? I mean, is measuring the interaction and classify all the interaction what I need to do in order to extrapolate what happens at a higher scale, right? That's, I think, it's it's not obvious, but I think it's better. I mean, I still think that there are. I don't know if that is the right experiment, but I think that there are experiments that you can do with just studying communities with two species that are useful to extrapolate. I don't know what experiments are, but I don't think it's like useless. Yeah.
And there was a point that you probably made about context dependency. So it is true. For example, when you think, okay, I want to measure this interaction and I want to call it some way. There's also the interesting thing is that many of these interactions are context dependent. So, for example, if you take resources, it depends on which resources you're providing. If you think of temperature, it depends on which temperature you are working. So again, deciding
I mean, for instance, in the simple example of glucose and acetate, right? If you then run the same experiment with acetate, you obtain a completely different type of interactions, right? Because one excludes the others, and that's it, right? So, yeah.
What bothers me is that differently from like the case when you study electrodynamics, and the evidence is ever increasing of how you can sum up things and they integrate well, and like the test particle you use to interact with the charged sphere and to produce a spark and like you all the variety of experiments people were doing when they were trying to understand electricity and magnetism were supporting the idea that you could somehow join those very restricted and partial experiments together. But when you have such diversity of results, vary in small conditions as you have as you seem to have in microbial ecology, I think it calls for a bit more care, a little bit more of caution when naming the things you measured and the results you got. Not that those results are not useful. They are, I think, very useful. Even the ones they are just pairwise. But then you should call pairwise things by their names and not by other names, which are so entangled with the ideas we already have from macro ecology, from the ecology of, I say macro not in the sense in the sense of big animals and the like, because then you get back to a terrain of a sanitized terrain where you can really not let all of the presuppositions you have from other areas of study come in and really have a more acute view, I think. So.
But look at on the other side. You want to communicate with ecologists, for example.
Okay.
So there is this problem that you are working with something new that maybe requires new names. But on the other side, you want to communicate with the people that have been doing these things with different species. So again, so there are always two sides. Those are good points, but there is the other side that if you want to communicate, you need to find common ground. And to me, for example, one huge problem that there is between, let's say, physicists working in ecology and ecologists is then they do not communicate because sometimes they call things with different names, even the same things. And so there is a tension of trying to define things because they are new, and sometimes there is the tendency of calling things that are old with new names. So it's communication in this sense it's there. If you want to bridge disciplines, and it's very complex.
I don't know if I really understood the question, but like for me, it's more a problem about familiarity with the term, the way you are used to use, than with the term itself, because like predation is a one-to-one relationship. It's a pairwise relationship between one individual and the other individual, and it's the way they are using here. And a positive interaction, okay, we have the biological concept of a +0 or a ++ interaction, but in a way, it's just something that when is interacting, makes a positive effect. And they are just using this for the community itself. When you see the community, there's a positive effect because if they wasn't together, they wouldn't have this amount of individuals. And the interaction of one-to-one, it's a plus minus. That is a parasitic one. So I don't know, the way the terms are being used looks for me that is more a counterintuitive thing because of the way we are used to use. and not the way it's used there itself. It's not a problem of the term, but the way we are just familiar to.
Yeah.
Well, I think that's a problem not only in this context, but in other papers from other kind of researchers, because it's not like if we should give new names, because there is that point that Martina was saying, but I think that the paper should state more clearly what they consider for each situation, what characterizes parasitism in this context, and other relationships. So I don't think that we need to use new names, but I think that's something related with what Joan was saying. But I think that they should just make a point characterizing what they mean by each interaction. That would help every reader, and it will be more clear for everyone. It's a problem that I was talking with Erica from other papers, because they use concepts like strains for some things and species for others, and sometimes you don't know exactly what they mean by the difference that they are seeing, if it's in a level of diversity. So I think it's something that we are missing in different contexts.
Yes.
No.
I mean, writing, so when you write a paper, you have, in principle, you should be really careful. But when we were here in discussions, the first things I said is, remember the way they measure the thing, because it's the measurement that defines then all these plus and minus, and then you can forget about the names, which was basically.
Even standard names like predation and parasitism, like there is the stereotypical scenario in which you have a small organism and a big organism, and that disparity in size and even in killing power makes you characterize that as parasitism, and in the case that you see a clear kill and you see a somewhat more balanced size relation, you say, oh, that is predation. But that is perhaps, I don't know the examples, but there probably are examples in which this disparity is not so clear. So you would say simply, oh, that is a negative positive interaction. So there is already a first step in which you give up the notion of predation and parasitism and simply talk about the positive minus interaction. I feel that microbial ecology poses further challenges which require giving up a little bit further some of the notions which we are attached to, because otherwise we are buying confusion with the notions, with the names we are already familiar with. So if you are in a context of a field which knows that, oh, sometimes I use signs to indicate things, and I have to be very attentive to what the signs mean in which context. Like, that's mathematics. Sometimes in the same area, you read a paper which uses a negative sign for something which is positive, which says something is 0 when it is indeed, for another paper, 1, because it established the metric in another way. So the field may someday, I think, be used to that and not be so attached to the idea of, oh, I have to determine whether it is positive or negative, and more and then be more attentive to because if you have a neutral ground in which you enunciate the results, then perhaps you can go back to those old terms and try and see them again in the.
I don't know. I don't think there is such a neutral ground, right? Because when you study microbial ecology, you study with a bag of knowledge and of concepts, which most of them they were developed not for microbial ecology, for what we're called macroecology, not macroecology, macro scale ecology, right? Macroorganisms, right? So it's clear that we focus a lot on competition, on we focus a lot on interaction types, etc., because we have this bag of concepts, which I don't think should be discarded, right? So I think it's all a question of balance, which it's hard to find. I mean, if you want a neat example of predation parasitism, is the case of phages, right? Phages are much smaller than the host. Phage means eat, right? So they are classified typically as predation, traditionally, but there are people that believe that they should be classified more as parasitic individuals, and we should use the same framework that we use for parasitisms instead the one of predation. So it's an example where this is like evident.
Maybe different pages have different mechanisms,
Yeah.
and so more complications.
Yeah. Yes. Very good. Well, thanks again. I mean, I think this discussion was yet another example of the reason why we enjoyed this. So these are the emails. Have you seen? So, if you want to be in touch in the future, we love that. Mm-hmm. So yes, if you have questions about this, or in general you want to keep us updated of your trajectories, we'd love that. Okay, thanks a lot.
Hey guys, yeah, I just want to give you a quick message. The lunch today with Thomas is canceled, okay? So please be here 2:30 for the class. We have the class 2:30. And now the picture.